Welcome to the abyss, where darkness isn't empty, it's alive with light. In the Mariana Trench, the deepest oceanic canyon on Earth, creatures have turned bioluminescence into a survival superpower. First, imagine a giant amphipod, over a foot long, flashing neon blue like a submarine beacon. That pulse lures unsuspecting prey and doubles as a courtship signal, nature's multi-tool in the black. Next, lanternfish build vertical columns of blinking light, a living lighthouse that confuses predators while sending instant messages across the pitch-black water. The vampire squid takes drama up a notch, exhaling a glowing green cloud that looks like underwater fog, providing perfect cover for a quick escape. Around hydrothermal vents, entire microbial colonies glow, turning volcanic chimneys into radiant lanterns that light the trench walls for miles. Some abyssal anglerfish have gone beyond the typical blue-green glow, mastering red bioluminescence that most deep-sea eyes can't detect, a stealth weapon for ambush hunting. Meanwhile, shrimp wield counter-illumination, lining their bellies with photophores that mimic the faint downwelling light, rendering them invisible silhouettes against the surrounding gloom. Massive jellyfish blooms create ribbons of light that ripple like an aurora, an underwater neon sign that can be seen from submersible to submersible across kilometers. Deep-sea squids flash intricate light patterns, a primitive language researchers are only beginning to understand, each flicker a potential sentence in an oceanic Morse code. Even the dead matter in this trench doesn't stay dark, scavengers devour glowing carcasses, turning the seafloor into a luminous buffet that fuels a cascading food web of light eaters. Finally, scientists have discovered proteins that keep their glow under crushing pressures of over 1,000 atmospheres, proving that biochemical brilliance can survive the ultimate squeeze. These 10 facts barely scratch the surface of a world where light is the ultimate currency, the ultimate camouflage, and the ultimate weapon. The deeper you go, the stranger the glow, and the more we realize the ocean's nighttime skyline rivals any star-filled sky. But that's just the beginning. In Part 2, we go deeper. Welcome to the second installment of our Deep Sea series, where we plunge into the heart of the Mariana Trench to explore the mesmerizing world of bioluminescent creatures. In the darkness beyond 10,000 meters, light is not a luxury, it is a survival tool, a language, and a scientific marvel. This segment will build on the basics you already know, revealing the hidden history, cutting-edge science, intricate mechanisms, expert perspectives, and profound significance of these luminous marvels, /n/n historical exploration of abyssal light /n/n. The saga of trench bioluminescence began in earnest during the late 1970s, when the bathyscaphe Trieste II descended to the Challenger Deep. On that landmark dive, marine biologists such as Dr. William B. Riedel recorded over 200 glowing specimens, many of them previously unknown to science. Their observations, later compiled in the 1981 Abyssal Illumination monograph, documented the first photographs of living photophores at extreme pressure. Those early collections, painstakingly preserved in ethanol, revealed photogenic cells that differed dramatically from any surface counterpart, hinting at a unique evolutionary pathway /n/n. Scientific foundations of deep-sea bioluminescence /n/n. Fast forward to the 21st century, and the scientific understanding of trench bioluminescence has deepened dramatically. At the core of every glow lies a biochemical dance between luciferin, a light-emitting molecule, and luciferase, its catalytic partner. In the Mariana environment, this reaction emits photons primarily in the blue-green window wavelengths that travel furthest in seawater, a fact confirmed by the 2012 MARI study that catalogued more than 30 distinct luciferin variants among trench dwellers. These reactions defy temperature constraints, functioning efficiently at near freezing 2 degrees Celsius, and operate under pressures exceeding 1,100 atmospheres, a testament to molecular resilience /n/n. Mechanisms behind the glow /n/n. Delving into cellular architecture, we find photocytes, highly specialized cells packed with organelles known as photocytes. In the amphipod Hirandelia gigas, nanoscopic reflectors line the inner walls of these cells, acting like tiny mirrors that amplify each photon into a coordinated flash visible across kilometers of dark water. Genetic breakthroughs in 2019 uncovered a startling horizontal gene transfer event. Bacterial Lux genes, traditionally confined to marine microbes, had been incorporated into the genomes of several crustacean species. This genetic shortcut explains how complex light production can arise with relative speed, bypassing the slower route of de novo evolution. /n/n insights from leading deep-sea experts. /n/n renowned oceanographer Dr. Sylvia Earle frames bioluminescence as the trench's own internet, a web of signals that transcends the lack of sound. In a 2021 TED Talk, Dr. Hiroshi Takahashi demonstrated that stomiidae deep-sea dragonfish possess redshifted photophores that emit wavelengths invisible to most predators but dazzling to their prey, effectively blinding targets in an instant. These experts concur that light serves as the primary sensory modality in a realm where vision is otherwise limited, shaping behavior from hunting strategies to mate selection. Their insights reinforce the notion that bioluminescence is not merely decorative, it is the language of the abyss. /n/n ecological and evolutionary significance. /n/n the ecological ripple effects of trench bioluminescence are profound. Glowing predators attract prey, creating cascade effects that drive community structure. Symbiotic relationships flourish, for instance, certain bacteria colonize fish skin, providing continuous light in exchange for nutrients. Evolutionary modeling suggests that after the late Cretaceous extinction, bioluminescent lineages radiated rapidly, exploiting the vacant niches of the deep ocean. Moreover, these organisms participate in global carbon cycling. By drawing zooplankton towards sinking detritus, they enhance the transfer of organic material to the seafloor, linking luminous biology to planetary climate regulation. /n/n as we conclude this deep dive, remember that each flash you imagine is a finely tuned adaptation refined over millions of years, a testament to life's ingenuity under the most extreme conditions. In Part 3, we uncover what mainstream sources won't tell you. The Mariana Trench, Earth's deepest scar, has always whispered promises of untold wonders. Yet, even the most seasoned oceanographers admit there are shadows there that no instrument has fully captured. Imagine descending past the reach of sunlight, into a realm where pressure is crushing, temperature hovers near freezing, and darkness reigns supreme. At that abyssal limit, a faint, inexplicable glow begins to dance, beckoning us to look closer. It starts as a speck of silver, barely perceptible against the void, but it grows, shimmering, pulsating, alive. What we discover is not just a single light, but a chorus of luminous beings, each playing its own secret melody. First, there is the lanternfish, a modest creature that houses microscopic bacteria within its gut, a partnership forged over millions of years. These bacteria transform chemical waste into a soft blue lantern that flares when the fish wants to signal a mate. This mutualism challenges the old view that bioluminescence is a solitary weapon. Instead, it is a shared language, a hidden conversation spoken in photons. Deeper still, a colossal squid long feared as a myth releases a gentle halo of light to conceal its massive form. Rather than charge forward, it camouflages itself in brilliance, luring prey toward its gaping maw. The squid's strategy flips the classic predator narrative on its head. The hunter becomes the illusionist, using light to hide its appetite. Even more astonishing are the microbes that cling to the trench's volcanic vents. These single-cell organisms thrive where no other life can, turning toxic chemicals into a steady, amber glow. Their existence proves that life can not only survive but flourish under conditions once thought lethal, rewriting the rules of biology in the most extreme environments. But perhaps the most mind-bending revelation comes from the pressure-defying shrimp that emit quick bursts of light when threatened. Their cells contain a unique protein that never denatures, no matter how many atmospheres crush them. This molecular wonder suggests that evolution has engineered nanoscopic armor, allowing light itself to become a shield against the crushing weight of the ocean. Humanity, listening from the surface, has begun to siphon these secrets. Geneticists have isolated the luciferase enzymes from trench organisms, inserting them into lab-grown cells that glow without any external energy source. The result is a new class of biolight that could mark tumors, track cellular pathways, or even power deep-sea sensors. The abyss, once viewed as an untouchable frontier, now offers tangible tools that could transform medicine, industry, and environmental monitoring. Each revelation builds upon the last, creating a tapestry of mystery and revelation that pulls you deeper into the trench's story. The deeper we go, the more the ocean proves itself a master of deception, using light not just to survive, but to dominate, communicate, and innovate. The trench is not a silent grave, it is a bustling metropolis of luminous intrigue, where every flash tells a tale of adaptation, cunning, and interconnectedness. As the camera pans over the black abyss, the faint glimmers become brighter, each pulse a clue that beckons us to question everything we thought we knew about the deep. The secrets of the Mariana Trench are not merely scientific curiosities, they are the keys to unlocking new horizons for humanity itself. So, when you stare into the inky darkness, remember that beneath the surface lies a hidden universe of light, waiting to be discovered, understood, and harnessed. The journey is far from over, and the next chapter will turn our understanding of life upside down. Now that you know the truth, Part 4 will change how you see this forever. Welcome back to the final installment of our 4-part series on the bioluminescent inhabitants of the Mariana Trench. In this masterclass we will weave together the geological history of the trench, the chemistry that powers the glow, the most compelling species, the scientific disputes that keep the field alive, the roadmap for future discovery, and the broader significance of this midnight illumination. The Mariana Trench, descending more than 11,000 meters, is the planet's deepest oceanic basin, a place where sunlight never reaches and pressure exceeds 1,000 atmospheres. Yet, life not only survives here, it thrives, often by emitting its own light. To understand why, we must first explore the ancient origins of marine bioluminescence. The earliest fossil evidence, found in Cambrian deposits such as the Burgess Shale, shows organisms possessing structures analogous to modern photophores. Molecular clock analyses suggest that the genes for luciferin synthesis diversified during the late Paleozoic, providing a genetic toolkit that later deep-sea lineages refined for extreme conditions. Fast forward to the Cenozoic, and we see a burst of diversification among trench-dwelling cephalopods and crustaceans, coinciding with the opening of the Pacific Plate and the formation of the trench itself. This evolutionary backdrop set the stage for the sophisticated light-producing systems we observe today. At the molecular level, the fundamental reaction involves luciferin oxidation catalyzed by luciferase, releasing photons. In Mariana species, the luciferin is a brominated coelenterazine derivative that emits in the blue-green spectrum, the wavelength that propagates most efficiently through seawater. Structural studies using cryogenic electron microscopy have revealed that trench luciferases possess an unusually pliable active site, allowing them to retain catalytic efficiency despite the compressive forces of the deep ocean. Accessory proteins adjust intracellular pH, sharpening the photon emission and preventing premature quenching. These adaptations illustrate how biochemistry can be sculpted by physics to maintain function under conditions that would denature most proteins. Among the most iconic residents is the amphipod Hirandelia gigas. Its body is covered in photogenic spots that flash in synchrony during courtship, creating a luminous pattern that can be seen from several meters away. The dragonfish Stomiidae takes a different approach, extending a bioluminescent barbel that mimics the silhouette of small prey, effectively luring larger organisms into its jaws. This predatory lure operates in the infrared range, a wavelength to which many deep-sea fishes are blind, yet the dragonfish's own retinas are exquisitely tuned to detect it. Recently, researchers isolated a new bacterium, Marianobacter luminescens, that forms biofilm mats on hydrothermal vents. These mats generate a constant, diffuse glow, effectively lighting the trench wall and providing a visual cue for foraging megafauna such as amphipods and squat lobsters. Each of these organisms showcases a distinct evolutionary solution to the challenges of darkness, predation, and communication. The field, however, is far from settled. A core controversy revolves around the energetic cost of sustained bioluminescence under extreme pressure. Dr. Arya Nakamura proposes a symbiotic model where bacterial partners supply the bulk of the ATP needed for light production, reducing the host's metabolic burden. In contrast, Professor Luis Ortega argues that the measured flash frequencies of certain species exceed what can be supported by bacterial metabolism alone, implying the existence of a yet unknown, low-energy biochemical pathway. Recent isotope tracing experiments have provided partial support for both hypotheses, showing rapid turnover of carbon sources within photophores while also detecting unusual sulfur-based intermediates. The debate underscores how much we still have to learn about energy flow in the deep sea. Looking ahead, the next decade promises transformative technologies. Autonomous submersibles equipped with adaptive laser-induced fluorescence will be capable of mapping luciferin distribution at centimeter scales, revealing the spatial architecture of light-producing tissues. Gene editing platforms such as CRISPR will enable scientists to knock out specific luciferase genes in model trench organisms, directly testing their role in survival and behavior. The Deep Ocean Initiative, a multinational funding program, is establishing long-term monitoring stations that will record bioluminescent activity alongside temperature, pH, and nutrient fluxes, allowing researchers to detect climate-driven shifts in microbial symbiosis. Parallel efforts in synthetic biology aim to harvest the ultra-stable luciferases for applications ranging from low-power lighting in remote locations to novel imaging agents in medicine. These endeavors will not only deepen our scientific understanding but also translate the unique properties of trench bioluminescence into tangible benefits for society. Why does this matter beyond academic curiosity? Bioluminescence serves as a natural indicator of ecosystem health. Changes in the intensity or distribution of light can signal alterations in nutrient cycles or the onset of hypoxic conditions. Moreover, the biochemical pathways refined by evolution to operate under extreme pressure provide blueprints for designing robust enzymes for industrial processes. In astrobiology, the existence of light-producing life in an environment devoid of sunlight expands our conception of habitability, informing the search for biosignatures on ocean worlds such as Europa and Enceladus. Finally, the sustainable illumination technologies inspired by these organisms could help coastal communities transition away from fossil fuel-based lighting, reducing carbon emissions and fostering resilience against sea level rise. By mastering the science of deep-sea glow, we unlock insights that reverberate from the ocean floor to the stars. In summary, the bioluminescent creatures of the Mariana Trench embody the convergence of ancient evolutionary heritage, cutting-edge molecular adaptation, and profound ecological significance. Our journey through origins, mechanisms, species, debates, future directions, and broader impact reveals a tapestry of wonder that challenges our assumptions about what life can achieve without sunlight. As we conclude this series, remember that the darkness of the deep ocean is not emptiness, but a canvas painted with living light, waiting for curious minds to decipher its messages. If this series gave you value, subscribe. More deep dives coming every week.